Students, you are welcome to your biology class, and today we are going to start chapter 6, molecular basis of inheritance. What is inheritance? Inheritance is the transmission of characters from parents to their offsprings, about which we have studied in our previous chapter in detail. Now we have also studied that how the characters are transferred and what is the genetic basis of all these informations, because we have studied about the Mendel and its laws. What the Mendel said, that there is something which is transferred from parents to their offsprings, and that there is something he named as factors. Means Mendel was not knowing much about the genes, so he said that the factors are responsible for transmission of characters from parents to their offsprings. Then after many years the discovery went on regarding what is this factor includes, or what is there in these factors which is responsible for inheritance. And then later on it was proved that there is a genetic material which is present in these factors and which are known as genes. And this genetic material is DNA, deoxyribonucleic acid. And in most of the organisms, DNA is the genetic material. And we also have studied about that what are nucleic acids. What are nucleic acids? We have studied in class 11th regarding biomolecules, that there are different kinds of biomolecules present in the body of living organisms, and one of them is DNA. And the other type of nucleic acid is RNA. Means there are two kinds of nucleic acids present in the body of living organisms. One is DNA and the other is RNA. Now, how these DNAs or RNAs are formed? So DNAs or RNAs are formed when nucleotides they are joined together. They form the nucleic acids. Means they form this DNA or RNA. So now, what is nucleotide? Nucleotide is a monomer. Nucleotide is a monomer, and when these nucleotides are joined together, they form the polymer. So, yani what we can say, that nucleotide is joining to form the nucleic acid, which is a polymer. Means when many of the units join together, they form the polymer. और कौन सी यूनिट्स यहाँ पर ज्वाइन हो रही हैं? न्यूक्लियोटाइड्स आर ज्वाइनिंग. There are two types of these nucleic acids, DNA and RNA. In most of the organisms, DNA is the genetic material, but in some of the organisms, for example viruses, they have genetic material as RNA. Now this RNA is genetic material in viruses, but it has some other roles in the body of other organisms also. For example, it can act as an adapter. Second, it can play the role of providing the structure. So there it is called as structural RNA. And in some cases, this RNA may act as a catalyst, and there it is called a catalytic molecule. Means RNA is genetic material in viruses, but it has some additional roles also. These additional roles are, it may provide structure, it may act as a catalyst, and it may function as adapter. Now, what is a nucleotide? Just we have studied that the DNA or the nucleic acid, DNA जो है, it is a kind of nucleic acid, and this DNA, it is made of nucleotides. So we will study that what is the structure of a nucleotide, because as we know that these nucleotides which are called as monomers, and they will form the nucleic acid which is called as polymers. So we will study about the structure of this DNA, or what is the structure of a nucleotide, then how the synthesis of DNA takes place, how RNA is synthesized. As we have talked that DNA is the genetic material in most of the organisms, but RNA is also present which is playing other roles. Now, how this RNA is formed in the body? This is formed on the template of a DNA, and that process is called as transcription. यानी DNA के टेम्पलेट पर DNA को बेस बनाकर जब RNA सिंथेसाइज हो रहा है, this is called as transcription. So we will study in this chapter about the structure of DNA, then how transcription takes place. Now when this RNA is formed, this RNA is responsible for the formation of proteins, and proteins जो हैं उनकी simpler unit that is amino acids. So what are the sequences of the amino acids in proteins, how proteins are synthesized? We are going to study about all this in this chapter. So first is DNA. Now DNA is the genetic material in majority of the organisms. DNA's full form is deoxyribonucleic acid. So it is a nucleic acid. Nucleic acid is made of nucleotides. So it is a long polymer of nucleotides. And when we talk about the length of the DNA, it varies from organism to organism, and it depends upon that how many numbers of nucleotides are joined together. कितने जो nucleotides join हुए हैं, जिन्होंने क्या बनाया है, वो polynucleotide यानी DNA. For example, a bacteriophage has 5386 nucleotides, whereas a human DNA, haploid content of DNA, it consists of 3.3 into 10 raised to 9 base pairs. In the same way, E. coli has 4.6 into 10 raised to 6 base pairs. So what we are able to see here, that there are different kinds of there is the difference in the length of DNA of different organisms, and this length is dependent upon the number of nucleotides attached. And when we talk about the structure of a polynucleotide chain, a polynucleotide chain, it may be of a DNA or it may be of a RNA. So first we will talk about the nucleotide. What is a nucleotide? अभी हम बात कर रहे हैं कि बहुत सारे न्यूक्लियोटाइड्स जुड़ जाते हैं और फिर वो एक लॉन्ग चेन बना देते हैं न्यूक्लिक एसिड की. सो व्हाट इज़ अ न्यूक्लियोटाइड? If we talk about the structure of a nucleotide, this nucleotide has 3 components. First component, it is, as we will see here in this diagram. Suppose we consider this as a nucleotide. ये एक न्यूक्लियोटाइड है. This is the second nucleotide, and what we are able to see that here, that these nucleotides, they are arranged, and this is forming when many of the nucleotides, means when many of the monomers, it is a monomer, this is also monomer, this is also monomer, and when these monomers यानी nucleotides जब join हो रहे हैं, it is forming a polynucleotide which is known as DNA, or it may form RNA. So we will study about the structure of a nucleotide, that a nucleotide, it consists of 3 components. This is the first we will study about the structure of nucleotide. This is the first component, we can say it is a pentose sugar. As we are able to see here the structure, it is pentagonal in shape, so, and it is a sugar, so it is called pentose sugar. Then what is the second thing present? G is written here. This G is a nitrogenous base, and P is written here. P is phosphate group. So what we are able to see here, that in a nucleotide it has 3 components: pentose sugar, this one, then nitrogenous base, and third is phosphate group. Now nitrogenous bases, as we are able to see here in this diagram, that at one place it is written as T, at another place it is written as A, here it is written as G, and here it is written as C, and all these are nitrogenous bases. So what we can say, that nitrogenous bases, they may be A, G, C, and T. Means there are different types of nitrogenous bases. Nitrogenous bases are categorized into 2 main categories, we can say. The first, it is known as purine, and the second category, it is known as pyrimidine. Purine and pyrimidine, these are the 2 types of nitrogenous bases, and these purines, they are further classified. They are of 2 types, adenine and guanine. So in short form, what we can write? A, or what we can write G for guanine. In the same way, pyrimidine, it is also of three types: cytosine, uracil, and thymine. And in the short form, what we can write, we can use the first alphabet, that is C, U, or T. Means nitrogenous bases are of two types, purine and pyrimidines, and they are further classified into different types. Now, as we are studying about the nucleotides are present in DNA also and RNA also. So what is the difference? This cytosine is common for DNA and RNA. हम क्या देख पा रहे हैं कि जो यहाँ साइटोसिन है, यूरेसिल है और थाइमिन है. साइटोसिन जो है इट विल बी प्रेजेंट इन DNA एंड इट विल बी प्रेजेंट इन RNA. लेकिन जो यूरेसिल और थाइमिन है, दिस थाइमिन इज प्रेजेंट इन DNA ओनली and uracil is present in RNA. So if it is given, if a picture is given or a diagram is given and where nitrogenous base is U is written, so you can easily identify कि अगर U यानी uracil, and if uracil is there it is a RNA. So this is the difference. अगर DNA है, DNA will have four types of nitrogenous bases, A, G, C, T, and if it is a RNA it will have A, G, C, and U. So this is about the different kinds of nitrogenous bases. Now a nitrogenous base when attached to a pentose sugar, as you are able to see here in this diagram also, this is a nitrogenous base, A, G, T. These are the nitrogenous bases. And what we are able to see, suppose we are taking this nucleotide as example. This is the nitrogenous base G. It is attached with the pentose sugar. और यहाँ पर क्या present है? One bond is there. So this pentose sugar and nitrogenous bases. यानी whenever a nitrogenous base is attached with the pentose sugar, then that nucleotide is known as nucleoside. Learn this. Definition may come in the exam, that what is a nucleoside? How will you define? When a nitrogenous base is attached to a pentose sugar, then it is called as nucleoside. And then the name, a nitrogenous base with this pentose sugar, and it is attached with a linkage or a bond, and the name of the bond is glycosidic bond. तो यहाँ जो bond है, this bond it is named as glycosidic bond and glycosidic linkage. और यहाँ पर जो न्यूक्लियोटाइड है इट विल बी कॉल्ड एज न्यूक्लियोसाइड. और फिर जो यहाँ नाइट्रोजिनस बेसिस हैं दे विल बी नेम्ड एज एडिनोसाइन. एडेनाइन की जगह पर इट विल बी कॉल्ड एज एडिनोसाइन. In place of guanine it will be called as guanosine. Then cytidine and uridine. So when this pentose sugar and nitrogenous bases they are linked by N-glycosidic linkage, then it will form a nucleoside, and the name of the nitrogenous bases will be adenosine, guanosine, cytidine or uridine. Now one more bond is there that is present between the phosphate group and the nucleoside. This one. This is the phosphate group. अभी इसका नाम जो है what we have said, this is the nitrogenous base. This is the nitrogenous base, this is the pentose sugar, and this is the bond which is known as N-glycosidic bond. अब इस स्ट्रक्चर के साथ में इफ पेंटोज शुगर इज ऑल्सो दिस फॉस्फेट ग्रुप इज ऑल्सो अटैच्ड and the bond which is present here, then this bond or this linkage is known as phosphoester linkage because it is present between the phosphate and the sugar. This is known as phosphoester linkage, and this phosphoester linkage, now it will be formed a corresponding nucleotide it is showing here, and two nucleotides are connected with each other. The name of the bond is phosphodiester linkage. Here you see this is one phosphate group, this is the second phosphate group. अब हम क्या देख पा रहे हैं कि जो दोनों ही न्यूक्लियोटाइड्स हैं, टू न्यूक्लियोटाइड्स, दिस इज सपोज फर्स्ट न्यूक्लियोटाइड, दिस इज द सेकेंड न्यूक्लियोटाइड. these nucleotides are linked together with each other, and the linkage इज थ्रू फॉस्फोडाइस्टर लिंकेज। यहाँ पर जो इनके बीच की लिंकेज है इन्हें हम क्या कहेंगे, फॉस्फोडाइस्टर लिंकेज। और इस तरह से टू फॉर्म आ डाइन्यूक्लियोटाइड। डाइन्यूक्लियोटाइड मीन्स टू न्यूक्लियोटाइड्स, यानी दो न्यूक्लियोटाइड्स के जो बीच में बॉन्ड प्रेजेंट रहेगी, दैट इज कॉल्ड एज फॉस्फोडाइस्टर लिंकेज। एंड इन दिस वे व्हाट वी आर सीइंग एबल टू सी हेयर कि एक पॉलीमर की चेन फॉर्म हो रही है। अब इस पॉलीमर में हमें एक चीज़ जो देखनी है, व्हाट इज देयर, that this polymer thus formed at one end has a free phosphate moiety, as we are able to see in the diagram. This is a polynucleotide chain, and in this polynucleotide chain, here this P is free. At one end phosphate is free, at the which end, 5 dash end of the ribose sugar, and that's why it is called as 5 dash end of polynucleotide chain. Similarly, at the other end, what is free? It has 3 dash OH, hydroxyl group is free, so it is referred as 3 dash end of the polynucleotide chain. Means a polynucleotide chain has two ends, 5 dash end and 3 dash end. At 5 dash end, what is present? Phosphate is present. At 3 dash end, what is present? OH is present. So there are two ends, and the backbone in a polynucleotide chain is formed due to sugar and phosphate, और इनकी जो backbone होगी that will be formed by sugar and phosphate, and the nitrogenous bases linked to sugar moiety project from the backbone. What we are able to see here, जो nitrogenous bases हैं, these are the nitrogenous bases. These are nitrogenous bases, and what they are formed, the nitrogenous bases किसके साथ attached हैं, sugar के साथ में. They are projecting from the backbone. वो backbone से कहाँ की तरफ project कर रही हैं, they are projecting from the backbone. So these are the structures present. So what we can say, backbone जो है it is formed due to sugar and phosphate. This is phosphate, then sugar, phosphate, sugar, phosphate, sugar. So backbone is formed by phosphate and sugar, phosphate and sugar, and these nitrogenous bases, they are projecting outside from this sugar moiety. So this is about the structure of a nucleotide chain, polynucleotide chain. Now if we talk about RNA, RNA is, we have studied that it has a different kind of nitrogenous base, and that is uracil is present in the RNAs. So in RNA, every nucleotide residue has an additional OH group. You have to remember this is the difference between DNA and RNA, that a RNA, what it has an additional OH group. This additional OH group will be present at the 2 dash position of the ribose sugar. यानी जो हमने स्ट्रक्चर ऑफ डीएनए पढ़ा वहाँ पर 2 डैश पोजीशन का कोई जो है देयर इज़ नथिंग अबाउट दैट, बट इन आरएनए राइबोस शुगर की 2 डैश पोजीशन पर व्हाट इज़ प्रेजेंट, OH इज़ प्रेजेंट, एंड व्हाट इज़ द नेक्स्ट डिफरेंस, दैट यूरेसिल इज़ प्रेजेंट एट द प्लेस ऑफ थाइमिन. थाइमिन नहीं देखने को मिलेगा, थाइमिन विल बी रिप्लेस्ड बाय द यूरेसिल. सो दीज आर द डिफरेंसेस बिटवीन डीएनए एंड आरएनए, कि एक तो जो है आरएनए चेन में इट विल नॉट हैव थाइमिन, इट विल हैव यूरेसिल एज अ नाइट्रोजनस बेस. एंड व्हाट इज द सेकंड डिफरेंस, दैट द पेंटोज शुगर एट 2 डैश एंड इट विल 2 डैश पोजीशन it will have additional OH group. Now DNA, as the name or as the full form is deoxyribonucleic acid. So it is, we can say, acidic substance. How it was identified and where it was first of all identified in the nucleus, and it was identified by Friedrich Miescher in 1869. So it is an acidic substance identified by Friedrich Miescher, where in the nucleus, and in 1869, and Friedrich Miescher named it as nuclein. But because of the technical limitations, the isolation of this DNA, it took a long time to study and isolate the DNA from the nucleus. And then in 1953, James Watson and Francis Crick, these are the two persons, This is the common model given by them. It is known as Watson and Crick model. And what they said and how they diffracted, they studied the X-ray diffraction data, और उन्होंने जो है फिर एक सिंपल स्ट्रक्चर प्रोवाइड किया, एंड दैट इज़ नोन एज डबल हेलिकल स्ट्रक्चर ऑफ डीएनए, एंड दिस इज़ द मॉडल विच टेल्स अबाउट द स्ट्रक्चर ऑफ डीएनए. So